the day. I know you had many other questions and there's been lots of discussion. So now is the chance for an extended deep dive into questions for our presenters. This panel session will be a mix. We'll introduce a wonderful new panel member, and then around about 2:40, Dean Miner, Dean of the School of Medicine here, will make his remarks for the symposium, and then we'll wrap up after that and move to the poster session. So let me hand over to Dr. Eshel, who will make the introduction of our new panel member.
Hi, everyone. My name is Eshel. I'm an assistant professor in psychiatry, and I'm taking over for Dr. Ruth O'Hara, who couldn't make it today. I'm also working with Leanne on the corporate affiliates program for the Center for Precision Mental Health and Wellness. So if anybody here is interested in learning more about that corporate affiliates program, please come find me at the break after this panel. So for our panel discussion today, we are incredibly fortunate to have with us Stanford neuro-oncologist, Dr. Michelle Monje. Dr. Monje is a professor of neurology and neurological sciences at Stanford and a trailblazer in oncology and neuroscience. Recently honored with the prestigious 2024 Ross Prize in Neuroscience, Dr. Monje has been recognized for her transformative contributions to understanding how tumors in the central nervous system interact with the brain. Her groundbreaking research has unveiled critical insights into how the brain's microenvironment influences cancer progression, revealing how signals from neurons can inadvertently facilitate tumor growth and spread. Dr. Monje's discoveries challenge established paradigms and reflect her unwavering commitment to advancing science and improving patient outcomes. So please join me in giving a warm welcome to Dr. Michelle Monje. So maybe I can just start the panel with a question for Dr. Monje. So as I mentioned, you are a renowned neuro-oncologist, but recently your work has taken you into psychiatry, and in particular, reward learning and opioid use. Can you describe a little bit how your work is already and can in the future inform our understanding of the pathophysiology or even the treatment options for mental illness?
Sure. Well, first of all, thank you for that wonderful introduction. It's really nice to be here. You know, I am in my core a basic neuroscientist, and I'm a pediatric neuro-oncologist trying to understand, you know, what processes enable childhood brain tumors to form in the developing brain. And I've always felt that the best way to understand that is to understand what is going wrong. So a lot of my lab studies normal neural development and neuroplasticity. And what we discovered a number of years ago is that the activity of the nervous system, of course, shapes neural circuits and affects the way that neurons function, but they do this not only through their effects on neurons in particular, but also on various kinds of glial cells. And so we try to understand these interactions between neurons and glial cells. And the childhood cancer that I focus most on arises from a precursor cell to glial cells, and actually a precursor cell to myelin-forming glial cells. And so we studied the adaptive and activity-dependent changes in myelination, not only during brain development and childhood, but also in an ongoing way through adulthood, and then how those really powerful interactions can be subverted in the context of brain cancers. And so on the non-cancerous side of the lab, we had been for many years studying neuronal activity-dependent changes in myelin and how those changes in myelin kind of tune neural circuit function to promote coordinated, circuit function, such as coordinated release of a neurotransmitter at their target. And we'd done this mostly in the cortex, but then I had this wonderful postdoctoral fellow who said, what about other neuron types? Might neuromodulatory systems be affected in this activity-dependent way? And might myelin change in a way that's important for psychiatric functions, not just cognitive functions, as we had been studying in the cortex and hippocampus. And so we looked at the dopaminergic system, which I think most textbooks told us was unmyelinated, but turns out it is well myelinated in many ways, and found that there was a region in reward circuitry that's very plastic, very adaptable to activity of dopaminergic neurons. And then found that plasticity of myelin was induced not only by sort of naturalistic activity of dopaminergic neurons, but also by drugs of abuse, and in particular by opiates, and found that these, activity-dependent changes in myelinated axons in response to opiates was required for opiate-associated reward learning because it tunes that circuit for coordinated dopamine release into nucleus accumbens. And so that's, sort of one direction whereby, beginning to study some interactions because of an interest in a cancer can lead us, through just following the science into studying diseases more relevant to psychiatry.
Thank you.
Yes.
Fascinating. Maybe ask the audience if they have.
Yeah. So we have questions prepared for each of the panelists, but it'd be wonderful to have it as interactive as possible. So please, if anybody is interested in asking a question to our new panelist, Dr. Monje, or our prior speakers, please feel free to chime in at any time.
And our speakers, if you have questions for Dr. Monje as well. This one here.
Well, since nobody else did, I will. One thing that I'm kind of surprised that no one has mentioned over the course of the 4 hours that we've been here, but is specifically related to neuro-oncology, is the role of the ketogenic diet, ketones, metabolic psychiatry. So I'm going to do my best to channel my inner Shabani Sethi. I'm on the advisory board of the metabolic psychiatry clinic here. To ask the question, what is the overlap between metabolic psychiatry and the role of altered metabolism in terms of treatment of depression and the anatomic pathways that we've heard about today? Where do they meet? Because perhaps understanding that might do better for both sides of the equation.
It's a question for Dr. Krystal.
I do not have anything to say on the subject.
I will speculate. So just to make it clear that I'm speaking in hypothetical terms. So we know that metabolism is critically important in regulating immune cell function and immune system function. And one of the consistent themes that has emerged as we've studied the way that neurons and various glial cells interact with each other, important for tuning, healthy circuit function, is that inflammatory state can really very dramatically change those interactions, even when the inflammatory challenge is subtle and not directly within the nervous system. And so one example of this is in, respiratory immune challenges. We found that COVID and influenza restricted to the lung can cause a microglial reactive phenotype that just regulates these neuron oligodendroglial interactions important for normal circuit dynamics and function. And so one could imagine that extending to psychiatric function and a range of different immune dysregulatory stimuli.
Take it back.
Andrew.
Andy Miller at Emory has been doing a lot of work looking at the effects of inflammation on brain. And it's not cancer, and I have no idea if it has anything to do with cancer, nor do I have any idea if it's relevant to metabolic issues and how they mediate effects. But it looks like his data suggests that it works at the nucleus accumbens and seems to cause dysfunction there and predispose people to anhedonia.
Given that it seems we've switched from metabolic or moved slowly along from metabolic to inflammation, some work that we've done in depression using DBSI, this is a diffusion neuroimaging measure that measures inflammation regionally within the brain. We found some associations in the big data set that I talked about before between inflammation in the amygdala and severity of depressive symptoms.
Well, no, it's very interesting that you mentioned that. Sunil Koliwad at UCSF has demonstrated hypothalamic neuroinflammation in response to a standard cafeteria diet, which gets better when you basically switch to a ketogenic diet. And
Right.
so we also know that standard cafeteria diet leads to gut inflammation, leaky gut, alterations in zonulins, allowing for transport of toxins, cytokines across the intestinal barrier, and the zonulins that make up the blood-brain barrier are the same ones. So one could actually, make a narrative. I don't know if it's true, but a narrative that actually this problem starts in the gut and ends up in the brain, and it's actually the same process. And that would actually tie these two things together.
Yeah, that's really interesting. We don't do a lot on the nutrition side of things, but we have just started a study looking at neuroinflammation as a link between late life depression and cognitive decline and risk for Alzheimer's disease. So that's something that I don't have any results yet, but I'm excited about.
Hi, really broad question, hopefully not too broad. But I'm just curious how you all think about increasing rates of mental illness. I think, a lot of us start our grant applications in this way, and you probably have experience and insight from your careers and just in terms of broad factors, societal factors, environmental factors, genetic factors.
Did he say race?
Rate. The rate of depression.
No.
The rates, the prevalence.
Psychiatry sort of.
Do you wanna have a-
So you're saying about the rate of depression?
Yeah, the increase in mental illness.
Right. Well, some of it's probably a recognition, right? Because COVID left us all at home. And so then when we came out, it was striking. I would expect it to go back to a base rate within 5 years. But I also think we're living in very chaotic times, not just the US, globally. And I think that can lead to people having, as I mentioned, more despair, just existentially around what's happening in their own lives. So that in and of itself, this is a broad remark, but could potentially increase depression. Whether there's an overall real increase in prevalence, I don't think I know the answer to that. Do you?
I think to Dr. Suppe's point is one thing I've seen in my clinical practice is that the world has always been a very chaotic place. And I would say actually historically has been more chaotic than it is today. But I see that my patients' awareness of the chaos is much more heightened for various reasons. And that plays in a lot of their despair. So just chewing on the problems of the world. I think we used to live in smaller communities with smaller circles and smaller awareness. And there's been great gains in opening up our worlds, but there's also some downsides to that. So not scientific, but one of the things I've seen.
And Leanne had the same data that I had, that the rates are going up. And from that data, I think the Lancet, it was because, folks are living longer. So there's more opportunity to have that go up. I think also we have the epidemic of loneliness. We are becoming less and less connected with each other in meaningful ways. There's a lot of, I think going back to the social media question, right? There's a lot of connection that they're not like as meaningful, as in the past when we were, I guess, in like smaller communities living together. So this epidemic of loneliness, I think, is also contributing to what we're seeing.
As I understand it, there's also been a very clear increase in the rate of autism over time that's not explained.
Hi, I just want to say this has been a great symposium, but I've also noticed we haven't talked about three things. We haven't talked about mania, we haven't talked about schizoaffective, and we haven't talked about schizophrenia. However, we've talked about some science that's just mind-bogglingly awesome. Do any of you have any ideas in the future of collaborating or even personally pursuing those topics?
You bring me the data, I'd love to.
I'm sorry, I couldn't quite hear you. What was your question about mania?
We've talked about a lot of topics, more specifically to the end of depression, PTSD. We've not talked about mania, we've not talked about schizoaffective disorder, and we've not talked about schizophrenia. What
Yeah.
you've brought up in terms of ideas, in terms of localizing the brain region, stimulation, possibly even with sensing,
Mhm.
blowing my mind right now, because while I find depression very interesting, I work in serious mental illness in an inpatient psychiatric ward. So
So I
we
have
see
a,
different stuff, but
right. Well, I have a 25-year history of studying bipolar disorder. So I care about it profoundly. And, bipolar 1 includes mania, but not bipolar 2. And there hasn't been a focus on funding except through pharma for about 20 years now with the change in NIMH, which has been a tragedy. But now there's beginning to be significant philanthropic funding to look at long-term course in bipolar 1 disorder anyway. And bipolar 2 disorder, because you can be medication-free more readily than in bipolar 1, I think bipolar 2 folks are actually, that it would be a great area to put more research into. I think you could do all the studies I was talking about and the imaging very readily with bipolar 2.
I was just going to say that I've heard also about the philanthropy investment increasing for bipolar disorder. So there's hope there. But another thought, mania is really hard to study. The people are very difficult to participate in experiments. That's one challenge. The other is the remarkable observation to me that we don't really have any idea why people switch state. And there isn't really even an animal model for like going from euthymia to mania or depression to mania. It's never been, and it's not been studied in humans. It's hard to predict. Like you can't put someone in the scanner and say, okay, now switch. We wish we could do that. But so there's some challenges there, but I'm hopeful.
I'm kind of a little reluctant to chime in on this one, but we do have a study that site PIing at VA Palo Alto. It's a VA-funded RR&D study where we're looking at different data streams to see if we can predict onset of these different clinical states, whether it's depression or mania, using voice recordings, using ecological momentary assessment, and using GPS. And so what we've seen so far is that there seems to be some just kind of in static signals that we can detect when somebody is more depressed because they're more in their house, with the GPS, not so much with some of the other things. But we have a big merit that's starting up. It just started in March.
And that's for people who are diagnosed, correct?
That's for people who are diagnosed. We can see when these changes are about to happen, because often, if you go from a euthymic state to a manic state, you might not recognize it, and you might be hijacked by it, and you might realize later, and then cycling back into depression. And so having insight into that and being able to maybe obviously prevent it from going full blown is what the idea is behind this. But it's very early on in this research.
And to piggyback off of that, you might look into Dr. Alu Ajalor's work. He's a collaborator of ours, and he's doing very similar work looking at applications. So a keyboard that's essentially installed into your iPhone or your Android device, and looking for markers of mood shifts.
Mind strong stuff, yeah.
I'll just add, there's been some really exciting, cellular molecular work, again, highlighting interactions between neurons and glial cells by Beth Stevens and Steve McCarroll. They published a paper a couple of years ago showing that microglia, which are the resident, innate immune cells in the brain, and one of their jobs is to prune synapses in an ongoing homeostatic way, that a schizophrenia associated genetic aberration in the complement pathway, which is one of the molecular mechanisms that sort of regulates the synaptic pruning, is associated with schizophrenia. And they've shown aberrant microglial synaptic pruning in humans with schizophrenia. So understanding that and trying to understand perhaps how to intervene might be one important pathophysiological component and opportunity for therapy.
thank you.
So question for Dr. Hilton. You talked about some work on guanfacine and the cognitive, biotype that I think was quite remarkable in a small sample. Could you tell us a little bit more about other precision trials using some of the other biotypes and other potential treatments to give us a hint of what's to come?
Absolutely. And I would like to just make sure, I am not a doctor yet. I just got candidacy, so hope to be. not there yet. Okay, so yes, we have done a little bit of work looking at the purple brain I was showing you, which is our positive affect circuit that we look at. We've had a couple of subjects that we've looked at matching pramipexole to, and so targeting the ventral striatum and sort of dopamine action, and that's been promising. We're hoping to extend that maybe into a larger trial down the road. We also had a recent paper that I believe the lead author was Leonardo Tozzi, recently come out, and it was looking at TMS. And so one of the things that we saw is that connectivity within the cognitive control circuit differentiated how people's cognitive performance improved. And so so we're trying out different things. We're selecting different treatments with different mechanisms, and I hope to see more.
So I'm wondering, can you hear me? So I'm wondering if you know of any projects going on about wearables and predicting. I mean, I would assume with heart rate, with other measures, you could kind of predict cycling, say bipolar, even, episodes of heightened schizophrenia, that kind of thing. Do you know of those kinds of things going on?
Questions about wearables.
There's a lot of work. There's a lot of work going on. There's a lot of research going on in wearables. I think a lot for assessment and ongoing. So there's a ton. I think you spoke to that a little bit. Yeah.
Yeah, Dr. Haijing Hollenbeck, who is in our lab, she is doing work right now. Who asked the question? Oh, there you are. Sorry. She is doing research right now with veterans, and this is called what's being called patient-generated health data. And so just even in the kind of the early phases of, would veterans, allow us to collect these kind of data, it's surveillance data, and would clinicians want these data, and how would they use these data, and how do you not overload clinicians with these data? And so I think it's some early stuff that's happening, at least in our lab, in this space with the Garmin watch and collecting these data to see if they might be predictive of, in her research, it's about PTSD, depression, and psychosocial functioning, so that there could potentially be a just-in-time adaptive intervention that could be deployed at the moment of need when the patient would be receptive to it. But we're just starting with that patient-generated health data and seeing if it's something that's of value to clinicians and if patients would be willing to use it, to collect it.
We'll come back to questions. We're thrilled in this segment to have Dean Lloyd Minor join us to officially support our symposium. It's my distinct honor and pleasure to introduce Dr. Lloyd Minor, the Carl and Elizabeth Naumann Dean of the School of Medicine and Vice President for Medical Affairs at Stanford University. As many of you know, Dean Minor has provided extraordinary leadership in shaping a bold vision to transform the future of life sciences at Stanford and beyond, with a focus on precision health powered by AI. Join me in welcoming Dean Lloyd Minor to the podium.
Well, thank you very much. I apologize for a little scheduling snafu this morning. I'd hoped to be able to be here and welcome Dr. Harris and also welcome all of you, but I'm glad I can do that just a few hours later than anticipated. But I want to reach out also first and thank Dr. Williams and Dr. Ruth O'Hara for their organization of this wonderful center and activity in precision mental health, and of course for this conference. Each of you is, of course, devoted to the research, the education, and the patient care associated with mental health and behavioral well-being and illness. And I think what's unique about Leanne's approach and the approach of colleagues working with her here and in so many other places as well is the focus on understanding, well, what are the biological correlates of both healthy mental health and behavioral health, and what are the biological correlates of, for example, the most common mental illness, the most common disturbance being depression. And for all to often, and if we look back on the precision medicine, precision health revolution, of course, the first impact has been in cancer. And today, a lot of the improvements in the outcome in the treatment of breast cancer compared to even a decade ago can be due to precision therapies and precision medicine. So a patient with breast cancer doesn't get a one-size-fits-all treatment. They get a treatment protocol and regimen that's determined based upon the tumor, the receptor status of the tumor, the overall health and well-being of the patient with the tumor. And as a result of not having one size fits all, but rather an evidence-based, evidence-driven approach to treating the specific tumor in the specific patient, we've been able to improve outcomes dramatically. And of course, that's one example, and there are many, many others. But today, in mental health and psychiatry, of course, we don't have the same level of precision. And because of the work being done here, and I know many of you and your other centers are doing comparable work, and it's so wonderful that you would share your wisdom and your expertise with us in symposia like this. But because of this work, we're hoping now not to be able just to say this patient has, in quotation marks, depression, but to be able to correlate the biology through a variety of different studies, both imaging, biomarker studies, as well as detailed analysis of responses to therapy, to be able to say this patient has this particular type of depression, and we know in an evidence-based way that this particular approach to therapy, whether or not it be a combination of cognitive behavioral therapy with medications or a myriad of other approaches, we know that that's the most likely benefit, will bring them the most likely benefit. But it's because of the work that Leanne does and her colleagues and their passion for understanding mental health and mental disease at a more mechanistic manner, for being able to correlate the biological correlates with what we observe in patients. It's because of this intensive effort that we're able to move this field forward. And of course, what we ultimately hope to do, just as with precision health, we hope to prevent breast cancer altogether. We also would hope to, through the studies like this, to be able to say, well, these are the correlates associated with the occurrence of depression. So if we had these particular modifications along the way, we would be able to lower the incidence of depression. We may be able to reduce the number of patients who have predispositions or correlates from actually developing a full-blown depressive disorder. It's a tall order, but I know that because of the work being done here and so many other places, that it's something that can and will be accomplished. So again, congratulations. Thank you for being here. And again, thank you, Leanne and Ruth and the panel here today for your dedication and your passion for what is truly, truly the opportunity and the need of our time. Take care. Thank you.
Question. Okay, so we can continue on the panel. Question for Michelle, Dr. Monje. So you are also an expert at the use of gene and stem cell therapies for brain cancers. As you look to the future for such therapies, do you see any opportunities for these approaches to be used for psychiatric disease?
Sure. So cancer cellular therapy has been really transformative for leukemia and lymphoma, and we've had some great success recently in treating certain forms of brain cancer with chimeric antigen receptor CAR T cells. And, you know, that has been incredible to see. One of the, I think, really exciting beyond cancer applications of CAR T cell therapy and other cellular immunotherapies is to treat autoimmune diseases. And we've just opened a trial here at Stanford, not me, but Jeff Dunn and others have opened a trial here at Stanford to treat patients with refractory multiple sclerosis with CAR T cell therapy, targeting those autoimmune, problem-causing lymphocytes. And that's an incredibly exciting application of this very, very specific sort of targeted immune intervention to sort of regulate the immune system essentially with itself, right? To engineer immune cells to re-regulate a dysregulated immune system. And as we understand more about a wide variety, I think, of neurological and neuropsychiatric diseases that have an immune component, there may be a similar opportunity to leverage these kinds of immune-based targeted approaches to normalize the immune system and therefore the nervous system. And so I think, there's a lot of increasing number of tools in our toolbox, and hopefully we can begin to apply them as we understand more about how immune dysregulation might be contributing to psychiatric.
So I had a
disease.
question actually about, thank you so much. So you mentioned multiple sclerosis. Do you see these approaches being useful potentially in other autoimmune diseases, which are fairly rampant, like SLE and so on?
Yeah, so that's the hope. So again, this is work, that I'm not personally part of, but that I'm, watching closely and really excited about. And I yes, these, there are CAR T cells, so engineered immune, cytotoxic T cells that are, designed to target a particular other kind of lymphocyte. And we first did this in the field for leukemia, but autoimmune, causing lymphocytes are another really important target. And I think there are now beginning to be trials for refractory rheumatoid arthritis, for lupus, for MS.
So would this be the kind of treatment you'd have to keep taking it?
What's
versus
amazing is no,
that's
because this is sort of a living medicine. So in the case, unfortunately, in the case of brain tumors, we do need to continue to administer CAR T cell therapy because of how immune suppressive those cancers are. But for leukemia, lymphoma, and hopefully for autoimmune disease, one dose of the CAR T cell enables engraftment of that, of that CAR T cell into the immune system of the patient, so that there's a continual living medicine. Now, these CAR T cells can misbehave, and they're generally engineered with an off switch. So there's the possibility to turn them off if they themselves begin to cause dysregulation. But I think it's a really exciting future direction.
Yeah.
But again, of course, we have to understand more about the basic biology.
Yeah, and what about circuit specificity? We've heard a lot about these various networks that might be involved, the biotypes that Dr. Williams, and Rachel, and almost Dr. had discussed earlier. Do you think, Dr. Monje, that your work will eventually lead us to this precision to a specific patient with specific circuit abnormalities, using maybe like the focus ultrasound technique that we heard about before?
I'm not sure if my work will lead us there, but I think work in the field certainly, and people at this panel are going to identify circuit level targets that can then be modulated, using really specific tools of, therapeutic tools of neuroscience. And I think that's incredibly exciting.
Question. Thank you so much. Great panel. It's nice to see the science. I've been coming to these meetings for a while, and it's nice to see the progression. However, my question is, TMS was mentioned a couple of times. And as a clinician, which is what I am, we have seen tremendous success with TMS in the past 15 years or so. I've been in the space and we've treated thousands of patients with success. And there was only a brief mention of TMS here. So I'm asking the whole panel their prediction of the future with TMS as a non-invasive treatment in treating depression and potentially other neuropsychiatric conditions. I'd love to see some talk, especially here at Stanford. Nolan Williams is leading accelerated TMS model, which is really revolutionary and 79 to 90% success rates. So I'd love to hear opinions from the experts here.
I think we have reason to believe that TMS will continue to evolve and improve as we optimize methodology. I think it's like any of the treatments going to be one among many. I don't think it's a cure-all. I think, for example, we're looking at new stimulation sites all the time and new frequencies. And, so there's a large parameter space to map, and that hasn't been mapped that well. So I expect incremental continued improvement. But again, it's just going to be one treatment among many. And I think, to me, it needs to be part of the broader system of when do you use which treatment for which person. And there's some who should get TMS probably maybe as their first treatment, and hopefully we'll figure out ways to know who they are.
Thank you for all the wonderful talks. So my question is, when you look at the trajectory of mental condition development, do you see there is maybe a critical or very effective time window to make the individual to be aware and receive treatment? Especially the data show that there's a significant lagging of people start to have symptom to the first time they receive treatment. Especially how would that impact being precise in terms of when, where, and how to interact with the individual. More important is like how to, especially some of the conditions may be also episodic. That's why the question is, how do you really take that into account when you're develop those different treatment strategies?
Just chime in a little bit about PTSD, where, yeah, the time from the trauma in civilians is like 8 years before they actually get any treatment. And in veterans, it's often, 20, 30, 40 years. The good news is this is the kind of what the data show is that that latency, at least for the PTSD symptoms, I'm talking about all the psychosocial collateral damage and other issues that come up with, family disruption and all the other things, at least for the PTSD symptoms, we're not seeing any interaction with how long it's been since the trauma on the PTSD symptoms using evidence-based psychotherapies. That may have changed in the last few years, but I haven't seen any data yet that, at least for the PTSD symptoms, again, there's a lot of other things that need to be cleaned up if you've had PTSD for 40 years, right? And but for the PTSD symptoms, at least that's what we say.
I think it's a great question. I wanted to echo Eric's response and tie it back to the TMS. Something that strikes us in the research is so many of the exciting new treatments that are available for depression, PTSD, other conditions, they're available only to people who have not done well on the other standard treatments. So you're hearing in our studies, oh, they tried 5 different treatments, or for TMS, you need to be defined as treatment resistant. So one of my goals would be that we can think of a way that these kind of assessments are available as part of the initial workup. And you're thinking, what is it at that time that could be suitable for someone early in the process, earlier toward the first instance? And that may be the case that then you're expediting either to TMS or maybe to some of the new psilocybin therapies. I think that's, to me, that's going to be really important if we want to make a dent. So we're not doing the trial and error. And maybe others want to jump in.
Yeah, just a thought about that. TMS is a great example. Why is TMS reserved for treatment-resistant patients? It's not because it doesn't work in people who aren't treatment-resistant. It's because it's expensive. And so we are interacting with economics, an economic landscape that drives clinical decisions in a way that maybe not be optimal. But we haven't presented them with the evidence that shows that this person should get this treatment, and if they do, the outcomes are better.
So I have a question.
Oh, I was just going to say, to Dr. Krystal's point, Dr. Laura Hack has started a precision psychiatry clinic at Stanford, actually. And one of the things I have learned to this point is that when we do generate that evidence and we present insurance companies with it, they're willing to play game. So we've actually had a few patients get reimbursed for having scans before we prescribe them medications. And so this is a real thing. If we continue on in this work, I think we are going to see changes in reimbursement and being able to get treatments first line.
One of the major reasons for advances in cancer therapy is early detection, the concept that we can predict high-risk people at risk for cancer before they get it. I haven't heard much about attempts for early prediction of mental illnesses. There must be a transition point between no symptoms and they're on the psychiatric ward. What kind of of studies are we doing for early prediction so that we can intervene in higher risk patients?
Thank you. Yes, I don't have answers yet, but we are doing a study along those lines using the ABCD data set. So this is another 10,000 participant data set of young adults. So getting enrolled when they're 9, 10 years old and then followed up every year. And so we're working at the moment developing a personalized kind of risk predictor based on both neuroimaging data, but also environmental and genetic and other data to try and predict their outcomes, both in mental health and also actually in cognition and sort of school achievement. So no results yet, but I think it's a really important question.
We can see childhood adverse actions and adverse events predispose people for certain cancers. I would be surprised if it didn't predispose people for mental illnesses. So we really, from my point of view, should be invested in prevention rather than treatment.
Absolutely.
I think if I could join, I think this is one of those areas where having a lot of data helps. So, you know, specifically within the All of Us program, thinking about, you know, prediction of diseases that have not yet occurred, but will in the future. And you actually don't have to wait that long. You can just sort of stop the clock at a certain time and what you know and use the information there to predict outcomes in the future and test that within data sets already collected. That said, I also understand there's a lot of confounding factors that aren't in data sets like the All of Us platform.
One quick, just to pique my interest, because my dissertation was on predicting PTSD from heart rate in the ER of the trauma survivor. And so with all of the wearable data that we're collecting, naturalistically, there's going to be lots of traumas. And if you could kind of see that trauma and see what, you know, when that happened, overlay it with the wearable data and kind of see what's happening with the clinical state. I think that would be a cool thing to do.
Related to the point made about the British data set. UK Biobank, that's the one. There's a group looking at the ABCD data set and looking at predictors of childhood depression from that multi-dimensional prediction. Another just tidbit I'll throw out is I work with some geneticists at UCSF, Louis Potachik and Yinghui Fu, and we've been studying genes related to sleep phenotypes and accidentally found that there's a gene for people who tend to need less sleep than everyone else that's protective against getting depression or PTSD kind of things. And this may help, and this is genes that are expressed in mice and studied in mice. We may learn something about circuitry that may be protective, which, you know, the mirror of that would be predisposition circuitry.
Yep. Have you looked at that in people?
Yeah, but
So
we can't study their brains very well.
That's the European, it's hyperthymia. So
Yeah.
it's not hypomania. And usually if you have folks like that, if you look up, down, or lateral, you'll find bipolar disorder.
We haven't seen that, interestingly. We have
Well, that's
what we
interesting.
have looked in their families
Mhm.
and somewhat extended families. And but these are rare mutations.
So they're with the genetic mutation.
Yeah,
not
an
just
autosomal
behavioral.
dominant,
Ah.
highly penetrant gene.
That's interesting.
Yeah.
I was going to say that on the flip side, there's also two researchers, a geneticist at Stanford and the vice chair, looking at resilience and how you can look at kids in Puerto Rico, swabs and send it out, potentially look at what makes people resilient, what are the genetic factors that are making them resilient, which I also think is an interesting flip side of actually promoting those protective factors.
Yeah, I totally agree. That's a key part of the project that I was mentioning earlier that we're working on.
So one of the things that struck me listening to all the presentations today on precision medicine is how we have, in one case, Dr. Harris creating an enormous database. You have Dr. Bijsterbosch using some of these enormous databases to tease out specific patterns. Then you have Dr. Krystal, who is doing in an individual patient basis, you know, or just a handful of 5 patients or so through a very intensive, deep, repeated kind of clinical trial. I'm just wondering if some of the panelists can discuss how we're going to bridge the gap between these enormous data sets and ultimately what we're looking for is these precision treatments for a given individual. Are they helpful to one another in some way, or are these done best in parallel?
I mean, I don't know that there is a gap. I think it is helpful to both sides. I read a lot of the precision literature, even though I do the big data kind of individual difference stuff. And I think kind of both ends of the extreme are very important, and hopefully will inspire one another. So I don't see it as a gap. I think it's very much complementary.
Yeah, I also don't think it's a gap. I think the ideal is getting very deep phenotyping, intensive data assessment in lots of people, ultimately.
Questions?
Yeah.
This fellow's been.
I do have an insightful question, but he's been waiting longer.
Yeah. Oh, thanks. So this was inspired by Dr. Suppe's talk, and I was just thinking about this phenomenon of rapid response, and then the fact that not everybody is able to seemingly consolidate those gains. And in psychedelic research and treatment, there are a lot of people who talk about this notion of integration. And similarly, I think when you look at CBT and medications for depression, you see differences when treatment is removed. And it got me wondering, are there individual differences in people's ability to consolidate the gains from after a rapid response to treatment? And should we be thinking about that as another potential area for individualization, say, for example, with a tailored digital intervention that could support people after they kind of break out of the rut that they were in?
Yeah, it's a good question. So there's work going on in natural language processing, both before and then after sessions, and they're looking for that exact kind of clue. So immediately after, what are the potential clues as to whether someone is going to show a no response, mild response, or a significant response? I don't think we have the answers to that yet. And again, it's part of why I like looking into the brain, which I don't think will necessarily give us all the answers, but it is a different look-see about what's going on. So there's momentary assessment data in this new study we're going to do with MDMA. We're doing natural language processing, momentary assessment, a bunch of stuff to try to understand better the trajectory of response and who responds and who doesn't. So it's a good question. Thank you.
And this is not necessarily an answer, but I think your question is very important because we want to prevent these future rapid acting, whether it be psilocybin, MDMA, turning into kind of ketamine, right? Ketamine came out on the market and it's kind of its application is a little bit of the wild, wild west, right? Very different models on how clinicians incorporate it and use it. And we don't want that to happen for these future therapeutics.
Hand in hand with the phrase precision medicine is the phrase digital twin. So but we haven't heard that today. I'm wondering the opinion of the panelists as to the overarching model for data analytics, rapid interventions and assessments, and modeling of us as a digital twin. And is there collaboration between your groups and that ecosystem?
I'm sorry, I'm not sure. I understand digital twins. I'm not sure I understand the question though. Can you?
Well, the question is, that's an initiative that's going on at a number of different places, but it hasn't been discussed here. And I was wondering if there is a role for putting the findings from this type of research into the broader digital twin ecosystem in healthcare.
I mean, I think it's a very interesting concept. What I've heard though today is, you know, in the randomized clinical trials that I've heard today, they tend to be fairly small sample, and so I think the digital twin initiatives that that sort of presumes that there are many patients or participants available, and so that would be my first thought is, would the type of research here at this point actually scale to be able to sort of have that take advantage of the digital twin framework?
I guess perhaps I'm wondering more about the predictive modeling and where this fits into building those models, and not just on an individual basis, but on a family basis and on a population basis.
Then I think definitely. So, I think that the big data sets that we're talking about, they're fantastic for hypothesis generation. They're also fantastic for predictive modeling. Again, we're starting to see studies that are done in one of the UK Biobank being replicated in all of us and vice versa. So I think that yes, predictive modeling and being able to sort of test that predictive modeling in various cohorts is definitely a win. Thanks.
So just to follow up on your question, Nir, I'm wondering, so for example, with these biotypes in depression, how quickly and efficiently can we use this information in a clinical context, right? Like, so how can we, do we have to brain scan everybody? Do we have to take everybody's blood? Do we have to have everybody's genome sequenced in order to do precision medicine? Can we do surveys? Can we do, what is the path to, actualization for research or in the clinic?
Comment about that. I think Leanne and Rachel probably have more insight about this than I do, but a perspective I have is that where I have tried to do relevant typing with clinical measures alone, it doesn't seem to be as good. It doesn't seem to work as well as the circuitry. And there could be many reasons for that, but I think that makes it hard to skip that and just let's give a person a scale and see if we can subtype them that way.
Yeah. Confirming that some of the work that we're just wrapping up is actually comparing different biotyping approaches in the same cohort to see to what degree they agree with one another in terms of the subtypes. And so we took 6 different biotyping approaches. 2 of them were clinical survey based, 2 of them were structural MRI based, and 2 of them were functional MRI based. And long story short, they don't agree whatsoever on biotypes, even though they each individually, I mean, the original papers were very elegant in terms of showing the biotypes. We can show some reliability internally and some validity internally within each approach, but there's no agreement across the different approaches. So that was part of the reason why the first study that I presented was more about kind of understanding the complexities in the different sources of heterogeneity, because I think there's a little bit more work to do there. But I'm not an expert on clinical integration, so I'll hand over to you.
Such a great question about how can we make all of this actionable, which was one of the themes of the meeting. I think one experience we've had is it really depends on the question that you're trying to answer. And if we're, at least in the case of depression, we found circuits are the most sensitive to predicting different treatment responses. And that kind of informs one path forward. So one of our innovations has been an image processing method, which will deliver back the biotypes in their current form to clinicians within about an hour. And that's something that Dr. Hack is using in her clinic. That's just one example. There'll need to be many others, including the digital measures, the genetics, and so on, and how to build the workflows for the first time in psychiatry, as they don't currently exist.
I was gonna, because I
Any
think.
final thoughts from our panel? Yes.
Well, it was just in your work, right, where you kind of could see it, where the person was having difficulty kind of forming sentences. And so there was clear kind of something there. And if you were to give them the guanfacine, so maybe there's sometimes when they're really hidden and maybe there's lots of interactive kind of things happening. But other times it's like, yep, there's a one-to-one correspondence. This clinic out in the middle of nowhere that doesn't have the ability to scan. In this case, go ahead and give them guanfacine, right? I mean, so it probably would be ultimately something like that, right?
Yeah. One, I think many of us are working on different angles of this same issue is how do you generate the mapping or a sense of surrogates in a way from going from circuit to what's the behavioral surrogate that you could measure, or the digital surrogate, if someone was not able to access the direct measure. And that is the same. I imagine that's got to be happening in other areas of medicine, right?
Yeah. Yeah, and so lessons from oncology, predictive biomarkers are key because that helps you stratify patients into appropriate treatment groups, do the right kinds of trials. We've learned over the last several years, tumors really are very heterogeneous, and you lose the signal of a helpful therapy for one subtype of tumor if you've lumped them all together. And so, kind of my field has spent the last decade or so using precision medicine approaches to try to appropriately categorize everybody so we can treat them better. And this is where I'm trying to find the person who asked the digital twin question, but where one hopes that something like a digital twin using data sets like those that are being generated by the people we've just heard from, in the coming years may be able to help stratify.
I think that's a wonderful note to wrap up on. I want to thank all of our incredible plenary keynote and guest panel members. So please join me in thanking them. Ahead of our poster session, which is coming up now, I do want to announce our prize winners and thank you to the judges. They were completely independent of myself. I am thrilled to announce the prizes go to, first of all, Divya Rajashekaran. Congratulations, poster 20. Jay Park, congratulations, poster 24. And Shui Chung, poster 19. Congratulations. Now, it's my pleasure to thank again the speakers and to thank all of you for attending today and generating such a wonderful discussion, excellent questions, ideas for future symposia and different topics that we can cover in the future. Please keep in touch with us. Please join the center if you're not already a member and share your ideas. We really look forward to the ongoing interactions. Please now